This is the third and final part in our series on ovulation, fertilization, and implantation. Ultimately, there are three distinct stages to implantation, and these correlate with the days after fertilization. So, after an egg is brought down into the uterus, it's still in the zona pellucida. It undergoes a hatching event where the trophoblast and the inner cell mass, the embryoblast, come out of the zona pellucida, and then it kind of rolls around on the endometrium within the uterus until it invades. So the distinct stages include apposition, adhesion, and invasion. We'll go over each of those one by one. In the first stage of implantation, which is apposition, the trophoblast releases enzymes and expresses proteins to facilitate proper erosion into the endometrium. Some of these proteins include adhesion molecules such as selectins that reinforce the rolling and tethering of the developing gestation to the endometrium, with the inner cell mass oriented near the implantation site. The endometrium also expresses proteins like MUC1, which is a mucin, which is an anti-adhesion molecule that prevents implantation at a relatively poor site. The next stage is adhesion or attachment. The mucin proteins need to be removed in order for successful adhesion to occur, so the trophoblast secretes enzymes that cleave MUC1 to promote attachment to the certain site. Attachment is also facilitated by a parade of chemokines and cytokines like LIF, or leukemia inhibitory factor, which exists to attract the embryo to a good site for implantation. Adhesion molecules like integrin attach the blastocysts to penopods, or mushroom-shaped cells in the uterus, only during implantation windows. These are the times when these cells are relatively receptive to implantation occurring. The third stage of implantation is known as invasion or penetration. This is where trophoblast cells from the blastocyst penetrate the endometrial epithelium, invading the underlying endometrial stroma with the purpose of reaching maternal blood vessels to provide a robust vascular supply to the developing gestation. The trophoblast cells develop invadopodia that grow adjacent to the endometrial epithelial cells and help really degrade into the endometrial basement membrane and allow the gestation to fully invade into the uterine tissues. All while this process is occurring, peripheral trophoblasts proliferate, differentiate, lose their membranes, and begin fusing with neighboring cells to form a sea of nuclei and cytoplasm called syncytium. Two trophoblastic populations now exist: the epithelial cuboidal trophoblast and the syncytial trophoblast. The syncytial trophoblast invades the luminal epithelium, which is another process called syncytialization. Syncytial trophoblasts are also those that release beta-HCG or gestational LH. The entry site of the embryo that is now fully invaded into the uterine space is plugged with a fibrin plug, and the syncytial trophoblasts become fluid-filled spaces that are separated by trabeculae. The trabeculae are arranged radially around the gestation, and cytotrophoblastic cells begin proliferating within the spaces, forming primary chorionic villi. Over time, the primary villi grow and branch into secondary and tertiary villi. Finally, as the trophoblasts grow, the cytotrophoblasts extend the villi into the endometrial stroma near the gestation. The nearby stroma then reacts to the invasion by swelling and filling with fluid with glycogens and lipids being released. This is called the decidual reaction. Once implantation occurs, the embryo breaches the luminal epithelium. The stromal cells that are surrounding the implanting embryo then transform into decidua in a process called decidualization. This is also facilitated by a number of immune cells, including macrophages and natural killer cells. So what else is going on while the embryo is invading the endometrial stroma? The inner cell mass is changing as well. The embryoblast subdivides into two cell types: cuboidal epiblast, most adjacent to the trophoblast, and flattened hypoblast that face the blastocyst cavity. The epiblasts secrete fluid as they proliferate, creating the amniotic cavity, and now the whole structure is called the bilaminar germ disc. Hypoblast proliferation extends around the blastocyst cavity, now forming something called the primary yolk sac. Midway through the second week, the embryo has completed implantation into the endometrium. A fibrin clot or plug closes the site of implantation here, and the gestation erodes further and further into the uterine tissues. It goes all the way until it reaches uterine vessels, which cause bleeding into the trophoblastic lacunae, which initiates the ureteroplacental circulation system. Cells from the trophoblast and the hypoblast continue to proliferate, and they undergo EMT, which stands for epithelial mesenchymal transition. These irregular mesenchymal cells have movement, and they expand into the space between the hypo and the trophoblastic layers, forming extraembryonic mesoderm. The trophoblast end up expanding faster than the hypoblast, and the chorionic cavity forms in between the yolk sac and the trophoblast. The trophoblasts continue growing, and they grow faster than the germ disc, and they continue to expand around the chorionic cavity. In discussing issues that might occur with implantation, thus causing some infertility issues, lift deficiency and integrin mutation are among some of the more common issues that might be seen here. LIF, if you remember, is one of the chemokines that is attracting the developing zygote to the endometrium. If there's a deficiency in this, you might see less likelihood of actual attachment in implantation. Similarly, integrin is another one of the proteins that's drawing the developing zygote towards the endometrium in order for attachment and invasion to occur. If there's a mutation in this specific protein, you will see less attachment and invasion and ultimately implantation.